Hey everybody, welcome back. In this exploration, we are going to be looking at some of the other patterns of inheritance that go beyond the simple Mendelian genetics. We've been talking about Gregor Mendel and how he did some research with pea plants, studying one trait with two alleles, a dominant and a recessive allele. But the reality is that most genetic patterns are not this simple. They usually feature some more complex factors. So that's what we're going to talk about today. And let's go ahead and get started. Okay. First of all, when we look at these different traits, most of the time we're looking at traits that are going to feature a various range of alleles. In Mendelian genetics, there's usually two alleles for one trait, a dominant allele and a recessive allele. But there's plenty of other examples where, maybe they don't have any dominant alleles. Maybe all of the alleles are recessive. Maybe all of the alleles are dominant and want to show up. There may be some combinations of some different things. So that's what we're going to look at here. First of all, incomplete dominance and codominance. I like to think of these two as two sides of the same coin. Incomplete dominance is where we don't have any dominant alleles. In incomplete dominance, the two different versions of a trait, if there is a heterozygous form, are going to create some sort of hybrid combination, some sort of middle ground. Like the example they give here is a white flower and a red flower resulting in pink flowers. The way that I like to think about incomplete dominance is if you mix together like flavors of Gatorade, like a red Gatorade and a yellow Gatorade. You mix those together and you're going to get some shade of orange. That is in contrast to the other type of pattern, which is codominance. And in codominance, this is a pattern where we have multiple dominant alleles. And if you remember, our definition for a dominant allele is that a dominant allele will always show up whenever it's present. Well, if you have two dominant alleles, then they're both going to want to show up. And what you get is this kind of like spotted or checkered or striped idea. So like right here, the example is a black chicken and a white chicken creating a black and white chicken. Now, the example that I like to use to help me remember what this one is like is a bowl of M&Ms. If you have a bowl of blue M&Ms and a bowl of red M&Ms, and you mix them together, well, you don't get purple M&Ms like you would if it was incomplete dominance, but what you get instead is just a bowl of red and blue M&Ms, where you can distinctly see the features of each individual bowl. And the same is true with this. In codominance, you can see the distinct features of each individual parent in the offspring because both of the different versions are going to show up because they're both dominant. Now, another wrinkle to this whole idea is the idea of multiple alleles. So to this point, again, we've been dealing with traits that have two different alleles. And even if those two alleles are either both recessive or both dominant, there's other options that are out there. For example, there's plenty of different traits where there are more than two possible alleles. Many different types of tulips, for example, have massive amounts of color combinations. There's not just two colors that they can choose from, like we saw in the pea plants. Another example of this in humans is blood type. As humans, we actually have three different alleles for blood type. We have what's called a type A, we have a type B, and then you might have heard it described as a type O. Now, blood type is a little bit tricky because there's actually this is a pretty cool combination of where you actually see incomplete dominance, codominance, and multiple alleles all at the same time, along with Mendelian genetics. So this is, blood type is kind of an example where you'll see multiple different types. Here's kind of a picture of it. Type A would have this marking on the blood cells. Type B would have this marking on the blood cells. The codominant form would have both of these antigens found, so you would see both A and B. And it's possible that you could have this type O blood with no markings at all. This particular allele is recessive to both of these first types, so like I said, lots of different combinations. We'll talk more about this example in class because it's a pretty good example of how to keep track of these different patterns. There's another pattern that can be found in traits that are located on the 23rd chromosome in humans, and these are called sex-linked traits. And the reason why these traits are different is because of the nature of how the 23rd chromosome works in humans. If you remember, in humans, the 23rd chromosome, we call it the X chromosome. If there are 2 full copies of it, then a person is going to have female physical sex characteristics. If there is 1 full copy and then this smaller kind of shrimpy piece called the Y, then that person is going to have male physical sex characteristics. And along with those physical characteristics, there are some other genes that are found on that X chromosome. For example, the ability to distinguish between red and green colors is found on that X chromosome. The gene that controls your likelihood of going bald is found on the 23rd chromosome. And this is why males are more likely to be colorblind and more likely to go bald, because they only have 1 copy of this. So if that 1 copy contains the recessive allele for colorblindness or the recessive allele for baldness, then those men are going to exhibit that particular phenotype. Females would have to have 2 versions of the recessive allele in order for them to exhibit these different phenotypes. So that's why you see many less females that are colorblind and many less females that experience baldness. It's possible, and they certainly could be colorblind or bald, but all a female would need is 1 non-recessive copy of that allele in order for them to be able to see color and keep their hair. So sex-linked traits are unique because the gender of the person matters in how that trait manifests itself in that phenotype. All right. Let's talk about a couple other factors that go into what our actual physical traits look like. Now, these aren't so much patterns of inheritance like the early ones are, but they're just an acknowledgement that most of our physical traits are more complicated than something that can be controlled by a single gene. For example, your height is controlled by many different genes. Scientists estimate that about 600 genes affect how tall you end up growing. It's not just a tall or a short. And in this case, we would call this a polygenic trait. Poly meaning many. So many different genes, multiple genes, contribute to the overall phenotype that you see. And this is a good thing because, obviously we don't want to just have a short gene or a tall gene, because that would limit the different possibilities of variation that we want to see in our population. Another factor in this is that the environment plays a role. The environment where we live, our diet, all those types of things can influence ultimately how tall we grow, for example. Another modifier factor is called epistasis. And we're not going to talk a lot about epistasis, but epistasis is an important idea that you need to maybe be familiar with, which is that one gene can modify how other genes are able to be expressed. And an easy one of these to remember is albinism. Albinism is a gene that, if turned on, will override the expression of other genes, for example, in pigment. So we have all kinds of genes that create pigments in our skin, in our eyes, in our hair. But if the albinism gene is turned on, or is present and is turned on, then that one gene can override all of the other genes that would contribute to the color being produced in our, like I said, skin, hair, and eyes. And that's called epistasis, where one gene can impact how other genes work. All right, and like I said earlier, the environment also plays a huge role in this. In humans, it's more about our diet. It's more about, our physical exercise, those sorts of things that influence, for example, our height, our weight. Other organisms, though, even something down to like the sex of the organism, can be influenced by the temperature outside. So Many environmental factors can also play a role in determining how those genes are expressed, how those genes are actually going to be seen in that organism's phenotype. It just kind of depends on, like I said, lots of things: temperature, diet, pH, even all these things can change. Two organisms could have exactly the same genetic makeup, but exposing them to different changes in their environment might lead to, like I said, variations in those physical traits. All right, we'll talk a lot more about this, especially as we get into probability and how we can predict these outcomes, but that brings us to the end of Exploration 3.